Today I'll be going to present a work done by these two people. Shubhadeep was a PhD student in my group, and he's a postdoc in Zagreb now. And Shashideep is a faculty member of BITS Pilani Hyderabad campus. So the work is motivated by cell biology, but it's a simpler motivation. So there has been recent work towards the direction of forming things which are similar to cell, artificial things, and as a result, much simpler than actual cells. So there has been recent surge of activity towards this direction. You'll see review articles, papers on things, keywords like synthetic cell, building synthetic cell. This is the project in Netherlands, and then artificial cells, etc. So you'll see these things pop up. Idea is that first thing we require for a cell is cell membrane. So one can start with some vesicle, and then if you incorporate some of the minimal amount of cytoskeletal materials in that, and then try to see one by one, if you add them, what do you see? What emerges from that? So, for example, this has been an earlier try on adding actomyosin cytoskeleton inside and outside a vesicle. So what you see is that if you add it on the outside, so initially this will generate a local structure, will generate deformations on the membrane, and if you keep on increasing activity beyond the point, it can slide down the cell structure and accumulate at one end. Or if it's connected everywhere strongly with the cell membrane, then it can crumple the cell from outside. And if it's inside, then also it can generate local fluctuations, active fluctuations, and local deformations of the membrane. And at very high activity, it can detach from the membrane, and the whole actomyosin contract, because of contractility, these guys come to the center. So, and there has been other attempts by other experimental groups. For example, Petrofield's group looked at cell membranes in presence of not cytoskeletal elements, but other proteins, which in presence of activity generates some interesting patterns on the membrane and shows some dynamical behavior. For example, they have identified things like pulsating membrane, so local pulsations, pole-to-pole oscillations, etc. Okay, so since this is a PDF file, I cannot play the movies, but otherwise we could have seen these oscillations. So what are the minimal things we are considering? So minimal things are like, I need a membrane. Membrane is modeled, we are doing analytical and numerical calculations. Membranes are modeled using surface tension for the membrane, bending rigidity, which is a Helfrich-like Hamiltonian, some volume constraint. And then there are activator proteins which sits on the membrane. So one can think about these proteins may have concave or convex shapes, and if they sit on the membrane, they will locally deform the membrane in that direction. And then they can move around on the membrane, and they do two things. One is that they can carve the membrane. Also, they can sense the membrane curvature locally. As a result, there would be accumulation of these things. And even these two ingredients, if I put together, that shows equilibrium patterns and has been studied by various groups, including this work, which I find to be very nice, Ramin Golestanian's work. And then we are interested in putting in the actomyosin cytoskeleton. So two dynamical non-equilibrium forces that will come from that. One is that actin polymerization, starting from these membrane-bound proteins, if they are activator proteins like VASP, from which actin can polymerize inward, which can control that, and that will generate an active push outward. And also these actin filaments, if they go inside the cell, in presence of myosins will pull them inward, and that can generate an actomyosin contractility, but that's in a different space, that's in the volume, and that will come to the surface via some propagator. So that's what we put in the model. Briefly, the model is that I take this membrane, on top of that I have this curvature sensing and curvature generating proteins which can diffuse on this membrane. There are certain cortical tethers, and then actin filaments which can grow and push this membrane outward. And then there could be actomyosin contractility, which can pull the membrane inward. So variables are this U, which is a local deformation from the sphere. And then there are two other variables we look at. One is this psi, the density field of this membrane bound proteins, and then there will be a field of phi, which will describe the actomyosin contractility. So one can write down these equations. This is equilibrium part, bending rigidity, surface tension, the tethering part, and this is the coupling of the membrane with the membrane bound proteins, that curvature coupling. And this is via this the Green's function, the contractility, the pulling force goes to the membrane, and then there is local pushing force in presence of the membrane bound proteins if actin polymerases, etc. So one can write down the dynamics for the deformation of the membrane, a coupled dynamics of the activator proteins, and a coupled dynamics of the myosin. So we are looking at small deformations around the sphere, so we can write this down in terms of the spherical harmonics, just expand in spherical harmonics and do the analysis, and one can do a linear stability analysis in the spherical harmonic modes, and one finds this particularly two interesting regimes which will generate patterns, non-uniform structures. One regime I am calling this U, so this is the regime of pattern formation, and then there is another regime here, so which is unstable spiral, which can generate both pulsation or localized running localized pulsation, which either localized or running pulsation. Yeah. So these are the results. Unfortunately, this movie will not play. So running pulsations run from one side to another. So this is some time snapshots of this guy. So as a function of time, you see that this is the structure which is changing. It was elongated this way, so it shrinks to the sphere and then again elongates that way in this specific example. So this behavior is like the pulsating structures seen in the experiments by Petroshal's group. And similarly, we get in another regime, we get to this running pulsation from one pole to other pole. Again, this movie will not play. And this running pulsation, you can see them in the theta direction, which is the polar direction of the sphere. The poles are chosen randomly by the system. And then one starts to see pole-to-pole oscillations of these things. And finally, if one thinks about, so these two examples where the membrane-bound proteins were generating outward curvature, and if I think about membrane-bound proteins which generates inward curvature, so in presence of that and in presence of the actomyosin contractility, it can generate instability towards formation of like local structures. So some simple ones are like L equal to 2 mode, which looks like the cell division. So all such structures will generate, and these structures are somewhat like similar to the structures seen in earlier theoretical work done by Ramin Golestinian's group. So in conclusion, I considered vesicle incorporating passive and active processes that shows pattern formation, running pulsation, localized pulsation, and these results, some of this behavior are seen in other experiments, and some of our predictions, particular parameter regimes, etc. The phase space can be studied in other future experiments where if one can have this clear control of the membrane and the actomyosin cytoskeleton inside it. So with that, I'll stop. Thank you for your attention.